Biology Part 23. Amino acid activation. Protein synthesis directed by mRNA can continue in this way, provided supplies of tRNA molecule carrying the appropriate amino acid remain available. The tRNA molecules needed are first synthesized in a molecule and have become linked to amino acids in the cytoplasm. This linking process is called amino cell and acid activation. It is also shown in the figure. The formation of a high energy bond between each tRNA and its corresponding amino acid. This bond energy is greater than the required for making the peptide band, and at the function of the peptide bonds within the ribosomes becomes a downhill process of RNA. The ribosomes do not allow it to appear there, and then we have got polyribosomes. Within limits, messenger RNA molecules can be reused because they pass through the ribosomes completely unchanged. The free end of mRNA molecules frequently becomes associated with a second ribosome before translation. A complete and depending on the length of mRNA can lead to the formation of this right content. Well, there are polyribosomes, and we have the control of the protein synthesis, and the process of protein synthesis described above depends on the transfer of the information within the cell. This transfer of information flow can be summarized in the form of a simple rule, and it can certainly bring great results. Well, DNA, RNA, proteins are the DNA being the internal conditions, and proteins having the external conditions. This pattern of flow gives feedback control of protein synthesis, which enables the cell to adapt and speedily, efficiently, or changing circumstances. The behavior of E. coli bacterial cells illustrates the principle very well. E. coli cells can be grown quite satisfactorily with minimum culture and mediation. With such culture is transformed into different medium containing the disaccharide sugar lactose in the cell. Soon begin the produced to enzymes called the biogalactosidase, which breaks the glycosides linkage between the subunits of lactose. Experiments of this kind show that the galactosides is only produced when it is needed, although the corresponding gene is always there. The gene for the enzymes is appropriate substrate, and it is illustrated as the first of these genes is called the repressor genes, produces molecules of RNA and synthesis. The repressor process in the absence of the suitable substance, in the case of lactose, is the protein binds and does not provide it over there. The cell's DNA operated gene region. They stop the sequence of the adjacent structural genes and provide the inducer repressor complex. This alters the shape of the repressor. Transcription, the structured lactosomes are there. The amount of lactose is available that starts in the eukaryotic organisms. The mechanism controlling genes repressing, and the organism is there. DNA different groups of genes can be expressed in each. These are the development proceeds of the different parts of organism. Some genes become permanently switched on while the others don't.